Hi there, this is Ms. Pearson, and I'm going to go over biotechnology with you. The Human Genome Project was done to map all 30,000 genes on all 46 chromosomes. This was done from 1998 until 2003, and the genome is the total DNA in the nucleus of each cell. So some fun facts about this project. About the genome, actually, there are approximately 3 billion bases, and the average gene is about 3,000 bases, but varies in size. And the total number of genes is estimated to be 30,000, and 99.9% of nucleotide bases are exactly the same in all people. And another fact for you, the project to map the human genome costs $3.7 billion. And in September of 2007, the complete sequence has been known of almost 2,000 viruses, almost 600 bacterial species, and about 23 eukaryotic species, half of which were actually fungi. So biotechnology refers to the technology to manipulate DNA, and the procedures are often referred to as genetic engineering. And then recombinant DNA refers to DNA from two different organisms. And this can be used for creating transgenic organisms, doing gene therapy, and cloning. So this was first used in the 1970s with bacteria. And to the right here, we have Dolly the sheep, which was cloned in 1996, and she lived until 2003. And it actually took 276 attempts before this was successful. Some recombinant DNA vocab for you. A plasmid, which we've talked about, is a small ring of DNA in bacteria. It contains genes which code for less essential traits like antibiotic resistance. And then a vector is a DNA molecule used to carry a gene of interest from one organism to the other. And restriction enzymes will cut DNA at a specific sequence. So if we look at that picture there, we've got our restriction enzyme and it cuts right here, and then we have now two separate pieces of DNA. So if we have a sequence of GAATTC appearing 3 times in this strand of DNA, it'll be cut into 4 pieces. So we end up with these things called sticky ends, the ends of the DNA. So how do we make recombinant DNA? Well, you cut the bacterial DNA with the restriction enzyme and cut the gene of interest from the organism's DNA with the same restriction enzyme. And then you combine the sticky ends of the 2 DNA pieces together with DNA ligase, which is also an enzyme. And this is known as gene splicing. So you'll insert the vector into a bacteria, and then the bacteria can now reproduce the recombinant DNA, and the foreign genes will be expressed in the bacteria. So if we take a look at this picture here, we've got our donor DNA on the left and our vector DNA on the right, and you can cut these pieces and put them together with DNA ligase and then introduce them into bacteria cells down here, and then the recombinant DNA will replicate and the cells divide, and you've got this DNA now from your donor DNA and your vector inside of this bacteria. So here's just another example of that. We've taken DNA actually from the bacteria and the nucleus of a eukaryotic cell, and now we can put together the plasmid DNA and the human DNA to do recombinant DNA. So there's some benefits of recombinant DNA in bacteria. Can make human insulin or human growth hormone, and the bacteria can actually be engineered to eat oil spills. So maybe you've heard about how bacteria was used to help clean up oil spills, like in the Gulf of Mexico and different places where that's taken place, where that's happened. So with genetic engineering, what can we do with genes? So we can use genes for DNA fingerprinting, doing an analysis of DNA sequences to determine one's identity. So we'll use electrophoresis to separate fragments of DNA based on their size. And how it works is gel with DNA is exposed to an electric field. and the DNA will migrate to the positive electrode because DNA is negatively charged, and the different size fragments will move through the gel at different rates. So the smaller the fragment, the faster it'll move. So here we've got our wells with the DNA molecules of different sizes in it on the negative end, and they'll migrate to the positive end, and our larger molecules will be closer to the wells, and our shorter, smaller molecules will move faster and go farther and be at the end there. There's also genetic testing, so you can look for the presence or absence of a particular genetic sequence, and you can look for genetic markers that are associated with various traits. You can also solve medical problems, so to be used to determine whether a particular person is the parent of a child. You can determine a child's paternity, so who their father is, or maternity, who their mother is. And the information can be used in paternity suits, inheritance cases, and immigration cases. So if we were to look at an example here, if you had a violent murder that occurred and a forensics team retrieved a blood sample from the crime scene and prepared a DNA profile of the blood sample, the victim, and the suspect, if we look at this picture here, can we determine who was a suspect? And well, actually, let me rephrase that. Was the suspect actually at the crime scene? So if we look at the suspect's profile versus the blood sample, you can see that these are the same, these two are the same, this is the same, this is the same, and this is the same. So yes, the suspect was at the scene of the crime. There's also gene therapy. So a normal gene is inserted into the genome to replace an abnormal disease-causing gene, may be used to treat a disorder by inserting a gene instead of using drugs or surgery. And types of gene therapy, there are either replacing or inactivating a mutated gene that causes a disease. So here's a nice picture that kind of wraps up some of those things that we've been talking about. So you've got the insertion of the therapeutic gene into this vector, and then the vector will attach to the surface of the cell right here and enter the target cell to incorporate the therapeutic gene into the patient gene, as you can see. So something else in biotechnology is cloning and CRISPR. So cloning is creating a genetically identical copy. So stem cells can be used to generate virtually any type of specialized cell in the human body. And the goal is to study human development and to treat disease usually when cloning happens. So this picture to the right is actually there are 5 monkeys here that are clones of each other, and in China in 2019 they did this to actually study Alzheimer's. So that's pretty cool, it's interesting. And then CRISPR is a specialized stretch, using a specialized stretch of DNA that produces a protein called Cas9, which will act kind of like a pair of scissors to cut strands of DNA, which is then used to edit genes. There are also GMOs, genetically modified organisms, so the insertion of DNA from one organism into another, or a modification of DNA in order to achieve a desired trait. And then transgenic organisms, they contain functional recombinant DNA. So here in this picture, we've got corn that's resistant to the herbicide Roundup, and on the right we have glowfish. So fish that have had their DNA modified so that they glow. Our common genetically modified foods are corn, canola, potatoes, tomatoes, squash, soybeans, flax, cotton, and sugar beets. And I put Fargo in there because there's a lot of sugar beets in Fargo where I went to school. And experts say that 60 to 70% of processed foods on grocery store shelves actually have genetically modified organisms. Ingredients, excuse me. So lots of things that we're consuming are genetically modified. So transgenic animals, they're usually put together, genetically modified for fast-growing foods, or chickens are more resistant to infections. They've modified cows to increase milk supply, have leaner meat, and produce less methane. And then like goats, sheep, and pigs, they produce human proteins in their milk and also this thing, biosteel. And so if we look at this picture of that salmon, you can see that the genetically modified salmon is much larger than the farm salmon, but they've been growing for the same amount of time. So they've genetically modified the salmon to put on weight much faster and be bigger so that they have a higher turnover of higher profit, I guess. You've got bigger fish faster. Transgenic plants, so can be disease resistant and insect resistant crops. So BT crops, they've got this bacteria that make a toxin against insects. It's a natural insecticide. And they'll also modify things to have hardier fruit, have trees that have hardier fruit or enhance nutrients. So in this picture here, you can see that actually biotechnology, genetically modified soybean is really popular. And these 4 down here are the most popular crops that are genetically modified. And they're mostly striving for herbicide tolerance and insect resilience, or the 2 combined. There are other uses of GMOs like biofuels and bioremediation. So we've got canola grown for biofuel, corn grown for biofuel. Poplar trees will remove groundwater contaminants, which is pretty cool. And we've got this EnviroPig that's able to digest and process phosphate. So there are advantages, obviously, and disadvantages to GMOs. So advantages, you can make crops that are like virus and disease tolerant. You can make crops with a higher yield, better nutrition. You can increase food security by changing the drought and cold resistance or increasing like shelf life in foods. Some of the disadvantages though is we're finding that more people are allergic to things, outcrossing or accidental cross-pollination. There are also super weeds, contaminating streams, biodiversity issues. There's also the question of the ownership of genes, like who owns Roundup ready crops? Roundup, I suppose. So what are super weeds? So we've got this plant here on the left, a regular crop or weed, and then the Roundup Ready one. So when you spray Roundup onto this plant, it's okay, it's resistant to it, but all of those bad things will get killed on the plant, where you sometimes have weeds that are actually not going to die when this gets sprayed on it, whereas this one, that's dead. This crop is okay, right? Because it's a Roundup Ready crop. So biotechnology pros: disease-resistant crops, new vaccines and medicines, bigger livestock for more meat, more milk, more wool, possible cures for diseases come out of biotechnology. You can make environmentally friendly organisms like that EnviroPig, the less methane producing of cows. There are also cons though, so it's unpredictable. The technology is new and there's really no guarantee that the products are free of side effects. It's also a money issue. Companies patent genes and demand high prices, and there are unexpected impacts on the environment. Biotechnology can be used as a biological weapon, and there are concerns over the safety and ethics of incorporating GMOs into food for human consumption. So then it comes down to ethics you got to think about. So like, how much do we have a right to know? Should they be putting non-GMO labels on foods? How much do other folks have a right to know about us? And like, if you do your ancestry DNA, do they own your genes since they have gone through and processed your saliva and stuff like that? So also, who has ownership of genes? Can a gene be owned? Can you own the genes of an organism? So lots of things to think about. Biotechnology is really cool and there's lots of stuff to study, obviously, but we went through that really fast. But if you have any questions, you want to learn more, ask your teachers, find some resources, they'd be happy to do that for you. All right, take care.